Topic 1.6, cell division. So first, we're going to be talking about the cell cycle and cyclins. Cyclins are family proteins that control the progression of cells through the cell cycle. Cells cannot progress to the next stage of the cycle unless the cyclin reaches a certain concentration. Cyclins bind to enzymes called cyclin-dependent kinases to activate them. The cyclin-dependent kinase attaches phosphate to proteins to become active and carry out tasks. So now we're going to talk about the cell cycle. The cell cycle is divided up into 3 stages. The first is interphase, then mitosis, and then cytokinesis. So the first stage of the cell cycle is interphase. This is the most active and the longest phase of the cell cycle. Here, the cell undergoes metabolism, endocytosis, exocytosis, and it obtains nutrients. So interphase is split into 3 stages. The first is G1, which occurs in the cytoplasm. This is where the cell grows and functions normally, undergoing everyday processes. There is rapid protein synthesis, allowing the cell to grow. The protein that is required for DNA synthesis is made, and the mitochondria and chloroplasts are replicated. Next is the synthesis stage, which happens in the nucleus. This is where the DNA doubles as DNA replication occurs, and the genetic material is duplicated, but no chromosomes are formed yet. And then the last stage of interphase is G2, which happens in the cytoplasm. In G2, the protein synthesis occurs to produce the proteins needed for cell division. An example of this is microtubule proteins that make up meiotic spindles. So the cell is actively preparing for cell division. The next stage of the cell cycle is mitosis. During mitosis, the nucleus divides into 2 genetically identical daughter nuclei, and the sister chromatids separate into individual chromosomes, which then get distributed among daughter nuclei. So mitosis is split up into 4 parts. The first is prophase. This is where the DNA supercoils, causing the chromatin to condense. The nucleus disappears and nuclear membrane disintegrates. The spindle fibers start forming, and the centrioles move to opposite poles. This is a picture of what prophase looks like. The next stage is metaphase. This is when the spindle fibers bond to the centromere of the sister chromatids, and they cause movement toward the equilateral plate. The sister chromatids are then aligned at the equilateral plate at the end of metaphase. So here's what metaphase looks like. Next is anaphase. Anaphase is when the sister chromatids are separated and pulled to opposite poles by the spindle fibers. This is what anaphase looks like. Last is telophase. During telophase, the chromosomes reach poles. The nuclear membrane starts to reform at each plate. The nucleolus appears in each new nucleus. The spindle fibers start to disintegrate, and the cell elongates in prep for cytokinesis. That's what telophase looks like. So to solve for the meiotic index, you can add prophase, metaphase, anaphase, telophase, and divide the sum of that by the total number of cells. The last stage of the cell cycle is cytokinesis. This is where the cytoplasm of the parent cell is divided between the 2 daughter cells. So in animal cells, there is a ring of protein located immediately beneath the plasma membrane at the equator that pulls the plasma membrane inward. This inward pull on the plasma membrane produces the characteristic cleavage furrow. When the cleavage furrow reaches the center of the cell, it is pinched apart to form 2 daughter cells. In plant cells, the Golgi apparatus forms vesicles that consist of material to build a new cell wall. The vesicles then merge and form the cell plate, and then the cell plate grows and divides into 2 daughter cells. So next we're going to be talking about tumorigenesis, which is the formation of tumors. Some key things to know are mutations, which are the change in an organism's genetic code. So when there is a change in the base sequence of a cell, it can result in a tumor. And some parts of the gene do not code, so mutations don't affect the organism. The next thing to know is what a mutagen is. So mutagens are the agents that cause gene mutation. Mutagens can be chemicals, high-energy ultraviolet light, shortwave ultraviolet light, or viruses. And the last thing that you should know is oncogene, which is a gene that has undergone a mutation that contributes to the development of a tumor. In their non-mutated state, they are called proto-oncogenes. There are two types of tumors, benign tumors and malignant tumors. Benign tumors usually are localized, they don't spread, and most respond well to treatments. Malignant tumors are cancerous growths. They can spread to other parts of the body, and they are often resistant to treatments. Some other things to know about tumors are tumors form as a result of a mutation in cyclins, CDKs, or proteins. Cells lose the ability to enter G1 and instead continue to divide rapidly, which passes the mutation onto the daughter cell. And last, once a normal cell division starts, a malignant primary tumor begins forming. If this is left untreated, this may lead to a secondary tumor.